Welcome to your first Neuroscience Laboratory session. This marks the beginning of a practical journey into understanding the structure and function of the human nervous system. The laboratory is not just a place to observe but a space to interact with real specimens, develop hands-on skills, and integrate theoretical knowledge with visual and tactile experience. In this introductory lab, our focus is on familiarizing ourselves with the organization of the nervous system, the handling of preserved brain and spinal cord specimens, and the identification of key anatomical landmarks. We will begin by reviewing basic laboratory safety rules and protocols for handling human specimens with respect and precision. The central nervous system will be our primary concern in this session, specifically the brain and spinal cord. You will be introduced to the external features of the brain, including its major divisions, the cerebrum, cerebellum, and brainstem. Begin by examining the cerebral hemispheres. Observe the gyri, which are the elevated ridges, and the sulci, which are the grooves separating them. Identify the major sulci such as the central sulcus and lateral sulcus, and use them to locate the frontal, parietal, temporal, and occipital lobes. Notice the two hemispheres are connected medially by the corpus callosum, a thick band of commissural fibers. Next, examine the cerebellum located posterior to the brainstem and beneath the occipital lobes. Its surface has tightly packed parallel ridges called folia. Observe the midline structure known as the vermis and the two lateral cerebellar hemispheres. The cerebellum plays a critical role in coordination, balance, and fine motor control. The brainstem, which connects the cerebrum and cerebellum to the spinal cord, consists of three parts: the midbrain, pons, and medulla oblongata. These structures contain important centers for respiration, heart rate, consciousness, and house the nuclei of most cranial nerves. Try to identify the cerebral peduncles, the ventral bulge of the pons, and the pyramids and olives on the medulla. Moving to the inferior aspect of the brain, identify the cranial nerve roots. Note that not all cranial nerves will be preserved in every specimen, but observe their general origin from the brainstem and their relative positions. Next, we move to a mid-sagittal section of the brain. This view allows us to identify deeper structures such as the corpus callosum, thalamus, hypothalamus, pineal gland, pituitary gland, and the ventricular system. The lateral ventricles, third ventricle, cerebral aqueduct, and fourth ventricle should be observed and traced. Identify the choroid plexus within the ventricles where cerebrospinal fluid is produced. You will then examine a cross-section of the spinal cord. Identify the gray matter in the shape of a butterfly in the center, surrounded by white matter tracts. The anterior horns contain motor neurons, while the posterior horns receive sensory input. Notice the central canal in the middle and the surrounding meninges, dura mater, arachnoid mater, and pia mater. Pay attention to the spinal roots. The dorsal root contains sensory fibers with a dorsal root ganglion, while the ventral root carries motor fibers. These roots join to form a mixed spinal nerve. Throughout the session, use anatomical models and prosected specimens side by side. Refer to diagrams, atlases, and your textbooks as you identify each structure. This is the time to reinforce your spatial understanding of the nervous system. You will also be introduced to the basics of histology in neuroscience. Under the microscope, examine sections of brain and spinal cord tissue. Observe the difference between gray and white matter. Identify neuronal cell bodies, glial cells, and large multipolar motor neurons in the ventral horn of the spinal cord. During this lab, ask questions, engage actively with your demonstrators and peers, and begin to build a three-dimensional understanding of the nervous system. This foundation is essential for appreciating the complexity of neurological function and for diagnosing neurological disorders in the clinical setting. In summary, this introductory neuroscience lab will orient you to the gross anatomy of the central nervous system, introduce key anatomical relationships, and begin to develop your dissection, observation, and clinical correlation skills. Let this session be a gateway into the intricate and awe-inspiring architecture of the nervous system.